Bio 139 lectures, blood. In this, we are going to learn about adaptive immune system. We are already done with the innate immune system. Now let's see about adaptive immune system. So our immune system, when it is intact and it is operating, it protects the body from infectious microorganisms, cancer cells, and the transplant and the graft cells. And it does this by directly attacking via cells and then also indirectly by releasing chemicals and by making antibodies. Now let's see what are the adaptive defenses. So these are our three lines of defense. So our innate defenses, the first one is the surface barriers. This is first line of defense and it consists of skin and mucous membrane. Then our second line of defense is this blue box, our internal defenses, and they include phagocytes, natural killer cells, inflammation, the process of inflammation, some antimicrobial proteins, and then fever. It is also second line of defense. So we went over all of this. Now in this video, we are going to see adaptive defenses. Adaptive defenses, they have a humoral arm and a cellular arm. The humoral arm, it is provided by the B cells, B lymphocytes, and the cellular arm, it is due to the T lymphocytes. So they are both lymphocytes. And there are some things which are important about the adaptive defense. Like I already said, it involves lymphocytes. And then it is specific. Our innate defenses, they were not specific. But this is specific. What does this means? That the adaptive defenses, they recognize and destroy the specific antigen that initiated the response. They do not attack any antigen. They only attack the specific antigen which initially provoked the response. And then this is systemic, right? Our innate defenses, like we talked, that inflammation, its purpose is to keep the pathogen and the injury localized. But this adaptive defenses, they are systemic. The immune response is a systemic response. It is not limited to the initial infection site. And then they have a memory. So after an initial exposure, the immune response is able to recognize the same antigen and mount a faster and stronger defensive attack. They do not forget. So these are 4 points which are important for the adaptive defenses. They involve the lymphocytes, they're specific, they're systemic, and they have a memory. Now let's see what are antigens. So antigens, they are substances that trigger the body's adaptive defenses, and then the body's adaptive defenses, they mount a response against the antigen. So antigen, it is actually a short form of antibody generating. So antigen is a substance which generates antibodies from the adaptive defenses. All right, so antigens, they can be proteins. Proteins are actually the strongest antigens. But then they can also be polysaccharides, lipids, nucleic acids. They can also be antigens. And usually the antigens, they are large molecules. Then we have another term. There is another term which is haptens. Haptens, they are incomplete antigens. So they are small molecules which do not they don't provoke the adaptive system to mount an immune response against them. But in the body, when they attach to other proteins in the host body, now that complex, it provokes the adaptive system to mount an immune reflex. So let's see what are the differences between the complete antigens and the incomplete antigens. So incomplete antigens, they are immunogenic on their own, whereas the haptens, they are not immunogenic on their own. They link to other proteins in the host body, and now the complex is immunogenic. They are both reactive. Okay, and then the complete antigens, they are larger molecules like macromolecules, proteins, large polysaccharides, some lipids, nucleic acids, and all of us have heard about the pollen response to pollen, allergy, and those things. So these are the pollen grains and microorganisms. They have these macromolecules in their structure, and because of that, there can be an immune response against them. Whereas these haptens, they are small molecules. Their size is small. It can be a peptide, a nucleotide, or a hormone. Sometimes some drugs, they can also be haptens. Only certain parts of an antigen, they can provoke an immune response. These parts are known as antigenic determinants. For example, consider this big antigen, and all of it is not immunogenic. Only certain parts, they are going to produce an immune response, and they are known as antigenic determinants. And different lymphocytes, they recognize different antigenic determinants. For example, now this molecule, it can engage three types of lymphocytes, colonies of lymphocytes to make antibodies against it. And variety of antigenic determinants, they're present on the surface of antigen. And then there are many large compounds like plastics. They have many identical, regularly repeating units, have little or no immunogenicity, and are used to make artificial implants. So that's why, because plastics, they do not have antigenic determinants. Otherwise, a regular molecule can have many antigenic determinants. And just to tell you that if there is one B lymphocyte and it is making antibodies, it will only make that specific type of antibodies. So now if this is attracting the B lymphocytes, it will attract three lines of B lymphocytes, and one will be making antibodies against this antigenic determinant. Then the second B cell line will be making against this antigenic determinant, and the third one will be making antibodies against this antigenic determinant. Okay, now what is major histocompatibility complex? It is written as MHC. Their self-antigens are the body's antigens that are not antigenic to itself, only to others. So our body cells, they have some self-antigens which are not immunogenic to my body, but they will be immunogenic to your body. And they are identified as self by MHC proteins on the surface of these cells. And it is unlikely that any two people, except identical twins, will have the same MHC proteins. And each MHC protein, it has a groove. So this is like I have made, so you this is a groove in this figure. This is where the antigen is going to attach, and then this MHC molecule, it presents the antigen to other cells, especially T lymphocytes. They need MHC molecules to recognize antigen. And there are two classes of MHCs, class 1 and class 2. Class 1 is commonly present on all the cells, and class 2 it presents the foreign protein. Now let's see how B and T lymphocytes, they are developed. So this is red bone marrow. Red bone marrow is the tissue where B and T lymphocytes are made, like rest of the blood cells. So these then B lymphocytes, these remain in the bone marrow, whereas T lymphocytes, they go to the thymus. That is why we put letter T with them, because they develop in the thymus. Thymus and red bone marrow, they are primary lymphoid organs. And when they become immunocompetent, they are called naive B cell and naive T cell. And what is immunocompetence? Immunocompetence, it is the ability of the body's immune cells to recognize specific antigens, and it is reflected by the presence of membrane-bound receptors. So now these naive B cells, they display certain receptors, and also these naive T cells, they display certain receptors. Those receptors tell us that they are now immunocompetent, although they are still naive. They have not been exposed to the antigen yet. And then what happens, that these naive cells, they go to the secondary lymphoid organs and they seed them. It is there in these secondary lymphoid organs that antigen is presented to them. So when they are presented with an antigen, they develop into a clone. So one cell, it mounts an immune response against one antigen. Okay, so now all of this clone, it is specific for this antigen. It will not produce any other type of antibody or cytotoxic T cells. If they are T cells, they are when they activate, they form cytotoxic T cells. And if they are and helper T cells, and if they are B cells, they produce antibodies, they produce cells that can produce antibodies. So those antibody producing cells, or that clone, it is now specific for this antigen. Now it cannot make any other antigen. So activated lymphocytes, they proliferate, multiply, and then differentiate into effector cells and memory cells. So they also become memory cells. Memory cells and effector T cells circulate continuously in the blood and lymph and throughout the secondary lymphoid organs. Okay, now let's move on to Let's learn a little about the development of T cells in the thymus. In the thymus, the T cells, they undergo two selections. One is known as positive selection, other is known as negative selection. What is positive selection? That the cells in the thymus, they present self-MHC to the developing T cells, and if they are unable to recognize the self-MHC, then they end in apoptosis. Remember, we talked about it. Apoptosis is programmed cell death. So in other words, death by cell suicide. The cell programs itself to die. So, and then those cells which can recognize the self-MHC, major histocompatibility complex, they are allowed to survive. And then in the second stage, there is negative selection. So again, the thymus, it presents the self-antigen to it, and those cells which bind very firmly to that MHC, this time they are eliminated. They are made to undergo apoptosis. Whereas the failure to recognize the self-antigen and bind tightly to the self-antigen, these cells are survived. They are allowed to survive. So that to make sure that these T cells will not mount an immune response against the self-antigen. It is just like that. You cannot be too lazy in a game and you cannot be too aggressive either. So you have to be perfect. So this is the end result, is that those clones of cells and those lines of cells are selected which will not attack the self-MHC. In the adaptive immunity, there are three important cells. There are B lymphocytes, right? Then there are T lymphocytes, and then there are antigen presenting cells. Antigen presenting cells, they do not respond to a specific antigen as lymphocytes do. And T cells, and then again the T cells, they cannot recognize their antigen without APC. So T cells are really spoiled. They need MHC to recognize the antigen, and then they also need an APC to present the antigen to them. Now let's see a little more into antigen presenting cells. These are antigen presenting cells, and this is MHC on top of an antigen presenting cell, and MHC has an antigen attached to its groove. So this antigen presenting cell is presenting this antigen to the T cell, and this T cell has a TCR, T cell receptor, on top of it. This is T cell receptor, and it interacts with the antigen, and after this the T cell is activated, and then we get two subgroups of T cells. There are those which have CD8 glycoprotein on their surface. They are cytotoxic T cells. They are the killer T cells. They kill now they kill the antigens and the cells which have those antigen. And then we have another subtype which is CD4 T cells. They are helper T cells. They help other B cells, they help macrophages, they even help other T cells. And then some of these CD4 cells, they are also regulatory cells, that they secrete certain substances to help the immune response. So this is how was a little thing about the activation of the T. There are three types of antigen presenting cells. There are dendritic cells, macrophages, and B cells. Dendritic cells, their only job in life is to present antigen, and they are present on the body surfaces that have contact with the external environment. And once they have the antigen, they are exposed to the antigen, they go to the secondary lymphoid organs, and then there they activate naive T cells. They present that antigen to the naive T cells. Then we have macrophages, and they don't ask for anything in return. That is it. They just present the antigen. Then we have macrophages, which are also antigen presenting cells, and they are present in the lymphoid organs and in the connective tissue, and they present antigen to the T cells to activate naive T cells and to maintain T cell activation. But they also have a motive. They are not like selfless like the dendritic cells. They also present the antigen to the T cells, so that in turn in return the T cell activation will also result in their activation. So they will be more activated. And activated macrophages, they are like killer cells. We know they eat everything. So then we have B cells, and B cells, they are like kind of selfish. They present antigen to the helper T cells for their own activation, right? So they're presenting to the helper T cell, so in return the helper T cell, it activates them. So our third line of defense, the adaptive defenses, and it has two arms, humoral arm and cellular arm. The humoral arm, it is because of the B lymphocytes which secrete antibodies, and the cellular arm is because of the T lymphocytes. So when the T lymphocytes, they are activated, they make CD4 T cells, which are the cytotoxic cells, and they make I'm sorry, I said it opposite. They make CD8 T cells, which are the cytotoxic cells which kill, and then they make CD4 T cells, which are the helper T cells, and they also regulate the immune response. And the B cells, when they are activated, they turn into plasma cells, and that plasma cell now makes antibodies. So let's go over the differences. B lymphocytes, they mount humoral immune response, whereas T lymphocytes, they mount cellular immune response. So the immune response because of antibodies, and the B cells, we call it humoral. And that because of T lymphocytes, we call it cellular, because T lymphocytes themselves, they kill. They don't secrete any antibodies or anything like that to kill the pathogen. They kill the pathogen themselves. And then the B lymphocytes, they secrete antibodies, yes. T lymphocytes, no. And then what are the primary targets of the B lymphocytes? They're extracellular pathogens like bacteria, fungi, parasites, some viruses in the extracellular fluid. Whereas the T lymphocytes, they attack the intracellular pathogens. For example, virus infected cells, cancer cells, where the pathogens are inside the cells, in the virus infected cells. And what is the site of origin? Both are made in red bone marrow. And what is the site of maturation? Like I said before, the B lymphocytes, they remain in the bone marrow, so they get the letter B, and the T lymphocytes move to thymus for their maturation, so they get the letter T. And what are the effector cells? Effector cells mean the cells which are actually doing the job. They are plasma cells. Plasma cells are the one which make antibodies. And for T lymphocytes, the effector cells are cytotoxic T cells, helper T cells. Then we have regulatory T cells. These are also CD4. And then memory cells, they both make memory cells. Yes. So we learned the basic things about the B cells and the T cells, and now let's learn about humoral immunity. Humoral immunity, it is because of the B cells. So this is a B cell, and the receptors on the B cells are antibodies, right? And when an antigen comes and it attaches to 2 or 3 or 4 antibodies, there is cross-linking of receptors happens, and this is what activates the B cells. See, this is cross-linking. So this antigen is attaching to 2 antibodies. And let's read from here. The immunocompetent but naive B lymphocyte is activated when antigen binds to its surface receptors. The activated B lymphocyte begins clonal selection, the process of B cell growing and multiplying to form an army of cells that are capable of recognizing the same antigen. So now it is going to grow and multiply, and all those cells that all that clone, it is only going to make antibodies against this antigen, right? So it has this has been like clonal proliferation. It is known as clonal proliferation of the cell. And then most cells of the clone develop into plasma cells, the antibody secreting cells of the humoral response. And the cells of that clone that do not become plasma cells, they become the memory cells. So these are our these lymphocytes, the adaptive immunity. It has a memory. So next time when the same antigen is going to attack those memory cells, they will take lesser time to become activated and to release antibodies and to get rid of the antigen. Now let's go over this. So this is the primary response. Primary response means the first encounter of the B cell with the antigen. So this is an antigen, and it has attached to multiple receptors which are antibodies on the surface of the B cell, and because of cross-linking, the B cell is activated. Now it is doing proliferation to form a clone. So these are activated B cells. So some of these B cells, they will become plasma cells, which will secrete antibodies, which will go and take care of this antigen which has attacked. And some of the cell, they will become memory cells, but they will now remember this antigen, and when this antigen attacks again, these memory cell, they will become a clone of cells identical to this cell, and then they are going to secrete antibodies. So the secondary response, it is going to be massive. It is going to take smaller time compared to the first response, and these memory cells, they can last for years and years. Now let's see, and the primary response. The primary immune response has a lag period of 3 to 6 days. So in 3 to 6 days, there are about 12 generations of B cells, and the peak levels, they reach in about 10 days. So this is the lag period. First, after at point 0, there was first exposure to the antigen A. Then about 3 to 6 days, the antibodies, they started appearing in the blood, and at day 10 they were maximum. Then they started to decrease. But when there was a second exposure to antigen A, and what happened, that this is the response. See, it is such a massive response that it only took 2 to 3 days, and this response is bigger, and it is more prolonged. It can last for months. So this is like response to another new antigen B. It is because the primary response, so it is a less smaller response. Now let's see what is active and passive humoral immunity. So humoral immunity, we know it is due to the antibodies. It can be active and it can be passive. What is active? Active means that which the body is making itself, right? So there is an exposure to antigen, and because of that antigen, the B cells, they make antibodies, and then they also make memory cells, and those memory cells, they stay in the body for long periods of time, and they remember the pathogen. So next time when that pathogen attacks again, the body, it has a very massive response to that pathogen. So this active immunity, it can be acquired naturally. How is it acquired naturally? You get some infection, and you get sick, but then you recover from that infection. Now your body has memory cells, and those memory cells, they are going to fight very hard when the same pathogen attacks again. So this is naturally acquired. And how is this active immunity artificially acquired? Through vaccines. Vaccines, they can be dead or attenuated pathogens. What are attenuated pathogens? Like they have been made not so that the body will not give a very big response to them. They have been somehow, slowed, or they have been humbled, you can say. So the vaccine, either it contains the dead organism, but that dead organism, why is it able to produce a response? Because the pathogens in its structure, they are present, and then they can be attenuated pathogens. There is some risk in the attenuated pathogens that sometimes they can become alive, like poliovirus is also comes in attenuated pathogen form, and sometimes it can those pathogens can become active. So I was talking artificially acquired is when through a vaccine. So this is what the vaccine contains. When we get a vaccine, they give us some dead organisms or some part of the organism or some attenuated pathogens, and those pathogens, they cause our B cells to turn into plasma cells and make antibodies. But the dose is because they are dead or attenuated, so the response is not very big. We do feel a little bad for 1 or 2 days, but we do not get sick. But what happens when next time that pathogen from the environment is going to attack us, our memory cells, they will have a very big a secondary immune response to it. So this is active immunity. It can be acquired naturally or artificially. Then there is passive immunity. What is passive immunity? That the pre-made antibodies to that pathogen are given to a person. And it can be naturally acquired. Yes, it also can be naturally acquired. How? The antibodies, they pass from mothers to the fetus via placenta and also in the breast milk. So they go to the infant, and now the infant has naturally acquired passive immunity, because we are not giving the pathogen to mount the immune response in the body. We are giving pre-made antibodies, and then this can be artificially acquired. So injection of exogenous antibodies are gamma globulins. They are given to a person, and so who has already become sick or with some disease. So then the shortcut is that they give an injection with pre-made antibodies. So this is active and passive humoral immunity. Now let quickly, I will not go into many details. So let's see about how the structure of antibodies is. Antibodies or immunoglobulins, they are proteins secreted by the plasma cells in response to antigen, and they are capable of binding to that antigen. So they have, this is their structure. They look like this. They have a heavy chain, and then they have a light chain, all of them. Okay, so and then they have in this portion, they have a variable portion. So this dark pink, this is the variable portion. So this is a variable portion, means that it will be different according to the specificity of that antibody. So this variable portion, it can change according to the pathogen. This is the variable portion. Let me draw here. This will be its variable portion, and this will be this antibody's variable portion, and then the rest is the portion which cannot be changed. So there are 5 types of antibodies: antibody M, antibody G, antibody A, antibody D, antibody E. So you can make a phrase, a word out of them to remember. I'm not since very creative, so I remember them like Magade, which is not very nice, but you can make a better word. So these are the 5 antibodies which our body makes. Now let's talk about cellular immunity. So cellular immunity is because of the T cells. T cells, when they are activated, they perform two functions. They become cytotoxic T cells, which have CD8 on their top, and they are the direct killing cells. And their targets are in the cells infected by viruses or bacteria, like we read in that chart, that intracellular viruses and or cancer cells, they are killed by the T cells. And then other foreign cells like in the transplant. So the transplanted cells, the T cells think that they are foreign and they attack it. And some of them become helper T cells and they activate B cells, macrophages and other T cells. And some become regulatory T cells and they release chemicals that regulate the immune response. They have CD4 proteins on their top. And now let's see about, let's go over this quickly. We will not go into much detail. So the things to remember are that our T lymphocytes, they require MHC molecules. They cannot recognize the pathogen without without MHC, and then they also want an antigen presenting cell to present them the pathogen. So they have some what can you say, they demand some protocol. So this is an immature lymphocytes, it is in the bone marrow, and then it moves into the thymus. In the thymus, T cell receptor appears on it, and then there is this this protein CD4 or CD8 is assigned to it. And now this goes to the secondary lymphoid organ, where this antigen presenting cell, this is a dendrite because this is what dendrite looks like, it is presenting antigen to it. So the CD8, so remember the number 8, CD8, the antigen is presented by class 1 MHC. This is how I remember 8. So 8 into 1 is equal to 8. Whereas CD4, the class 2 MHC presents the antigen to it. So 2 into 4 is equal to 8. So this is how the CD4, they are activated by the class 2 MHC, and CD8, they are activated by the class 1 MHC. And then there is colony proliferation, just like it happened with the B cells. And then CD8 cells, they become cytotoxic T cells. And then the CD4 cells, they become the helper T cells or the regulatory T cells. Just a few words about the MHC proteins. So MHC proteins, they have two groups. There are class 1 proteins and class 2 proteins. Class 1 proteins, they are expressed on all the nucleated cells of the body. Our RBCs are not nucleated, so they do not have class 1 MHC proteins. And then the class 2 proteins, they are expressed on the antigen presenting cells, the dendritic cells, the macrophages, and the B cells. And then this class 1 is recognized by the CD8 T cells, and class 2 is recognized by the CD4 T cell. Remember the number 8. 1 into 8 is equal to 8. 2 into 4 is equal to 8. And then this class 1 antigens, they have the endogenous pathogens. Okay, so the endogenous proteins. So there can be intracellular pathogens or proteins made by the cancerous cells. Alright, and whereas the class 2, they have the exogenous proteins like phagocytized extracellular pathogens. When some pathogen it is engulfed, and then its protein is expressed from the class 2. So this is how our this were just a few words about MHC, and now we are done with the meeting.